Hello and welcome. My name is Isabelle Schiffer. I have a PhD in genetics of aging, and I'm here today to speak in my role as scientific spokesperson to you for the Forever Healthy Foundation. And the talk today will be about how close we are to having senolytics as a rejuvenation therapy for humans. But before we go into that topic, I want to introduce the Forever Healthy Foundation for those of you that don't know us yet. Forever Healthy Foundation is a private nonprofit initiative with the mission to vastly extend our healthy lifespans. And we want to be as healthy at 70 as at 30. And while that has been a silent revolution, the world without age-related diseases is coming closer and closer. And the groundwork is done and the research is accelerating. And drugs and therapies are not only under development, but the first generation is already available today. So it's really not an if, but a when. And it also the same for senolytics, but there has been a lot of premature excitement, and the first senolytic treatments being in clinical trials now moved to the market, and it has been marketed in a cowboy style market, how we like to call it. So it's really marketing driven service offerings with no transparency, no trusted source of information, and no focus on risks and risk management. And we want to provide guidance in that regard, and we use a certain approach. Our approach is that our team of medical doctors and scientists screen all related scientific papers on a certain treatment. We evaluate the relevant preclinical and clinical studies, and we then assess the risks and benefits. We call this risk and benefit analysis, and then publish these results open access. And why do we do that? Because we want to enable application. We want to have that information available for free for everyone on our knowledge base, so that people that want to use a certain therapy already, or medical doctors that are curious about that, find the information about the risks and the benefits. And with that, we always try to be really early, and for senolytics, we are quite early in the field to provide this information for everyone who thinks about using these therapies already. So we are really providing high quality guidance, but we are also increasing global awareness of, yes, we're moving forward, but we still need to be cautious. And with that, we're jumping into the topic, senolytics from bench to bedside. Talking more about the bedside today. Where are we now? And first of all, we of course need to talk about senescence, because this is what it is all about. And the title is The Good and the Bad, because I want to strengthen again that senescent cells are not only bad, but senescent cells have a biological implication. So the healthy cell experience acute stress, for example, during a wound or an injury, then this is acute stress, and then senescence, it can turn into a senescent cells, which is beneficial. It's biological function to be a senescent cells, and also the secretory phenotype here, this molecules that are excreting are really important for this specific task. And then afterwards, they are going to be cleaned up by the immune system. And so the biological function that we know of today is that it's really important in wound healing or in development. What we talk about when we want to develop senolytics is the chronic senescence, which is happening when there is a gradual increase in stress or damage. Then also cells can turn into senescent cells, which are not cleaned up by the immune system. And then they turn into late senescent cells, which is happening during aging and age-related diseases. There are certain hallmarks postulated on senescence, which is senescent cells are in a cell cycle arrest, so they are not dividing anymore. They're resistant against apoptosis, which is quite peculiar, because mostly they upregulate signals that should actually drive them into apoptosis. But at the same time, they ramp up anti-apoptotic pathways that I show you later, that keep them alive. And also one of the hallmarks is SAS production. SASP stands for senescence associated secretory phenotype, and it's mostly inflammatory cytokines that can harm the surrounding cells and cause tissue damage. And what are the milestones that are relevant to senotherapy? Because I think this is really interesting to show, because whereas the Hayflick limit is shown 9061, for those of you who don't know, the Hayflick limit is a cell divides for a certain amount of time and then it just stops. And it was shown that this is due to a shortening of telomeres. Telomeres are these protective caps that are sitting on the end of the chromosomes, and with every cell division, they are getting a bit shorter. And at some point, they're critically short, and then the cell stops dividing. And then 30 years later, markers were identified to really identify senescent cells, which is, for example, beta-galactosidase or P16. P16 is also currently used for genetic removal of senescent cells and an inducible system in mice studies, which is quite elegant. But quite interesting that in the 90s, the markers were identified that we can really identify what's the senescent cells, or the first markers at least. And then in the 2000s, it is shown that senescent cells somehow are involved in aging, because they are found in higher numbers in aged mice, but also in primates and in human atherosclerosis, difficult word. 2008, the SASP that I told you, the secretory phenotype of the cells, what it really is, was then shown in 2008. And in 2016, so only 4 years ago, it was shown that killing of senescent cells extends the health span and lifespan of naturally aged mice. Before, it was already shown that removing senescent cells from premature aging models in mice also rescued some of the age disease aging defects. But I think this was really an important study to show that by killing senescent cells in naturally aged mice that were then better, it really has an effect on aging. And then in 2017 also, it was shown that senolysis is shown to have a therapeutic potential in the treatment of osteoarthritis, and it's shown to be also implicated in fibrosis, and that made it now possible also to move into clinical trials, as you need to have some sort of disease that you can also be treating with this therapy to be moving to human trials. And this is now done. So we have the first clinical trials that came out in 2019 for Dasatinib and Quercetin, which I tell you a bit more about later, which is quite exciting. But I just want to emphasize here that the milestones were only in the recent years that really drove senolytics now to clinical trials. And what are senolytics? How do they work? So first of all, they should be eliminating senescent cells in a targeted fashion. So they should be eliminating senescent cells, but not healthy cells. And most of the senolytics that are present at the moment target anti-apoptotic pathways. I told you in the introduction that senescent cells really ramp up anti-apoptotic pathways to stay alive, and those senolytics target these pathways in specific. But there are several different ones, and here on the right side I depicted that there is, for example, BCL2, P53 or P21, HSP90, and several more. And it's shown that senescent cells upregulate, different types of senescent cells upregulate different of those anti-apoptotic pathways, whereas the senolytic mostly targets one of those. So the current senolytics are quite cell type specific. And there are two that are very promising. And this is why we decided to do a risk and benefit analysis of them. Both are in clinical trials already. For one, that we have results, for the others we're still waiting for the results. I'm gonna say we just jump into the analysis right now. So first, I want to introduce Fisetin. Fisetin is a quite promising senolytic. Both senolytic as well as Dasatinib were identified in a screen where molecules were identified that inhibit senescent cells or deplete senescent cells, and Fisetin is shown to target really to eliminate targeted senescent cells. And it's quite promising because it's a natural senolytic, and this is why now it comes the strawberries on the right side. Like strawberries have a high amount. Strawberries have a high amount of Fisetin. So this is why it's maybe thought to be less potent. Natural compounds are usually less potent, but probably it also has less side effects. So it could be more readily translatable into a therapy. So we set out to assess what is known so far about the benefits and about the risks. And the benefits are quite striking. So it is shown to inhibit this GAP, so this anti-apoptotic pathways in vitro. It induces apoptosis of senescent cells in a quite specific way. In animal studies in mice, it is shown that it reduces markers of senescence and it increases the lifespans in mice and in flies. Unfortunately, there are no results on the clinical trials so far. There are, I think, two or three clinical studies running at the moment at the Mayo Clinic. And we might get some results this year, which would be quite exciting, but so far there are no results. Concerning the risks, Fisetin as a compound is a very safe compound. Only in very high doses it showed liver toxicity in mice. However, I want to emphasize here that little is known about the side effects of senolytics in general, especially in humans. The possibility is that there might be a wound healing defect. As I told you in the introduction, senescent cells are important for wound healing, so there might be a defect in wound healing or fibrosis. We don't know that yet. And to reduce these risks, there are strategies, for example, to use senolytics in an intermittent dosing, because it has been shown that they work longer than the time you take them. So for example, the protocol that is currently used in the Mayo protocol, because the clinical trials are running at the Mayo Clinic, they use 20 milligram per kilogram on 2 consecutive days and then they stop. And it has been shown that the senescent cells are being targeted way longer, so beyond the treatment. And this could be a way to also reduce the side effects if you don't need to take this for a longer period of time, but you still have the effect. Yes, so for Fisetin, it is quite promising so far in animal studies. Again, there are no results in humans yet. So the second senolytic therapies that we reviewed were Dasatinib and Quercetin. Dasatinib is a cancer therapy, so that's why during our screen we found more than 3,000 papers, because it's intensively studied. There are many publications because it's really for years used in cancer therapy. It also came out of the screen that I told you about, the initial screen, what is targeting senescent cells. And quercetin more falls in the category of Fisetin, so it's a natural compound. And it's shown that Dasatinib and Quercetin tackle different senescent cells. So as a combination therapy, it's supposed to be quite powerful. We found from these over 3,000 papers, we found that 150 qualify for our analysis. It's preclinical, but also clinical studies. So there are 3 clinical studies where they use Dasatinib and Quercetin as combination as a senolytic therapy. And then we identified at the end 8 benefits and 56 risks. To go a little bit more into detail, the benefits in animals are shown in different organs, as I told you, and the combination therapy of them both might be then able to target several different kind of senescent cells, and there is an effect shown on different organs, and it looks quite promising. However, most of these benefits are shown in disease model, for example, in diabetic mice. And again, here it would be interesting to see what the treatment does to healthy aged mice. And furthermore, 5 of these 8 benefits are blood markers, which are at the moment what we use, so blood markers for senescent cells, but we don't really know how it really translates into a clinical relevance. Concerning the risks, there are numerous risks that are known for Dasatinib because it's used as a cancer treatment. So most of the risks that we found were, of course, from these papers, and the risks correlated with dose and duration. And as I told you before, it is thought that it could be used intermittently because it works longer than you take it. So you could use it for a short amount of time and still have the benefits days or weeks later. So this could be a way to reduce the side effects also. So that's why we went back to our analysis and checked when do the first side effects occur. So could we use it for a shorter time? And several of these side effects occurred even shortly after initiation. So at the moment, we would not recommend as a healthy person to use Dasatinib and Quercetin. So there are 3 senolytic Dasatinib and Quercetin trials, which are phase 1. They are not representative at the moment, but they're really crucial to have. They're done on a small group of patients with idiopathic pulmonary disease and chronic kidney disease, and they show beneficial effects, which is quite promising. However, the risks that we found are quite heavily, so we should not, we should wait for more clinical trials to show really that Dasatinib and Quercetin is also a safe treatment to use. And with that, I want to summarize a little bit the current state on senolytics for rejuvenation to use in humans. Despite the side effects, so there is some promising, there are really promising therapies. However, now coming from regarding it from the human side, there is really no consensus on measuring their efficacy. So for blood samples, so in humans, you need to have something that you can really get easily, so it needs to be blood, and you can measure blood markers, but do they really translate into clinical relevance? And this is one concern. There is no coherent treatment protocol for humans, which will be, of course, in the next clinical trials more clear what is the best treatment protocol. But so far, there are no studies that really show clear benefits in humans. And again, because this now sounds all quite negative, I want to emphasize again that this field is really promising, but it's just a very young field. So we just moved in the past years really towards showing that senescent cells really have an effect on aging, and now we could show that it also has therapeutic relevance for age-associated diseases, and the first clinical trials are there. We just need to be a bit more patient. And what we also think is quite important to develop at the same time. So first of all, we need to have short and long-term effects. We need to know the side effects in humans. We need to have a treatment protocol, but also a therapeutic window. So when do we start treating people? If we want to use it as a rejuvenation therapy, when do we start treating people? Is it in their 20s, in their 30s, in their 50s, in their 70s? The therapeutic window needs to be defined. there also need to be maybe a strategy which is more aimed at this late senescent cells, so that we don't hit the good senescent cells, which is all, of course, under development. And furthermore, we need specific and sensitive biomarkers. As mentioned before, there are some biomarkers that you can see in the blood, so senescent markers, but are they really specific? Are they really sensitive? And how do they translate into clinical relevance? And last but definitely not least, we think we need to understand more about senescence. So there is now more and more known about the biological function, and there are studies published now that show, for example, one recent study by Dimitri Bulavin, who showed that constantly removing senescent cells also have detrimental effect, because in some tissues they might have a function, or they at least are crucial to be there. So it also might be detrimental to remove them. So we need to work further on the strategy, and I think research is going strong, and the field is really accelerating. Just at the moment, there is now senolytics yet for humans, so we just need to be a bit more patient. And with that, I want to end my presentation by saying that we are looking for new members of our team, and maybe with this presentation you could already have a grasp of what we are doing. So we are really being early on, reading the newest advances in the field of rejuvenation and writing this risk and benefit analysis. So if you want to join our team, then please apply and visit our foreverhealthy.org career site. And we also have different other risk and benefit analysis that you can find on our website. And with that, I want to say thank you for your attention.